Okay, thank you, Dr. Hu, for introducing me, and thank you everyone to come to listen to our talks. So, yeah, today I feel very glad to have this opportunity to present you my work on brain sciences and bipolar disorder. So, we know that bipolar disorder is a very serious and debilitating psychiatric illness, which is characterized by extreme fluctuations in moods and disturbance in thoughts and behaviors. So according to the World Health Organization, around 40 million people worldwide suffer from bipolar disorder, and it is a leading cause of disability across the world. So bipolar disorder reduces the lifespan of patients by over 10 years, and it's very important to note that this condition is very difficult to detect and diagnose, which results in many individuals with this condition to remain undiagnosed and untreated. Because of this, it's very critical for us to have novel and objective brain markers which we can use to complement clinical assessment and to improve the sensitivity of detecting bipolar disorder. So in this context, we need to consider two very important characteristics of this illness. So firstly, this disorder has a known biological basis involving changes in the brain structure and functions. And secondly, bipolar disorder is a highly heritable disorder, which means it runs in families, with offspring of bipolar patients carrying much higher risk of developing this disorder compared to people without family history. So today I will introduce three aspects of my research to you. And firstly, we discovered a novel brain feature which contributed to the identification of individuals who already developed bipolar disorder. And second, we uncovered brain features which characterize high-risk individuals who are offspring of bipolar patients. And thirdly, we directly assessed early brain markers which prospectively predict high-risk individuals who later develop bipolar disorder versus those who remain clinically well. These studies collectively carry clinical implications for early identification, prevention, and intervention of bipolar disorder. Okay, so I will now first introduce my first aspect of work, which was to discover this novel brain index which contributes to the identification of bipolar disorder patients. So we know in the human brain, different regions, they're interconnected to form networks which perform important mental functions. On the structural level, different brain regions are connected via white matter tracts that can be visualized through structural brain imaging. On the functional level, we can infer the connectivity between different brain regions through computing the statistical correlations of their activities over time. Existing evidence shows that bipolar disorder patients may show abnormalities in both structural and functional brain connectivity, but they may also show abnormalities in the association or coupling between structural and functional brain connectivity. The coupling between structural and functional brain connectivity is a very important index because it reflects how well the brain utilizes the existing physical connectivity structure to achieve well-coordinated interregional activities in a functional domain. To address this question, in one study we compared 57 bipolar disorder patients against 42 matched healthy controls on both their structural and functional brain connectivity, as well as their coupling between structural and functional connectivity. So here is the analysis pipeline and major findings of this study. So for each individual participant, we computed both their structural and functional connectivity matrix. And the results showed that, as expected, so compared to healthy controls, bipolar disorder patients showed significant reductions in both structural and functional connectivity index. But most importantly, they also showed significantly reduced coupling strength between structural and functional connectivity. And when all the three indexes were considered together in the same model, we found that only this coupling index between structural and functional connectivity emerged as an important brain feature contributing to the identification of bipolar disorder patients. So this study provided novel evidence supporting the use of the coupling index between brain structural and functional connectivity to help with the identification of bipolar disorder. I will now introduce the second aspect of my research, which was to uncover brain features characterizing high-risk individuals. So in this study, we focused on the particularly vulnerable individuals who are offspring of bipolar patients and who already showed some subclinical bipolar symptoms. So using machine learning technique, we constructed a classification model using the brain gray matter volume data, and we obtained a model which successfully identified those particularly vulnerable individuals at an accuracy of over 80% and sensitivity of over 90%. In another study, we followed the high-risk offspring of bipolar patients by over 10 years and discovered that those offspring of bipolar patients shows a different pattern of brain development during adolescence compared to the low-risk controls. I will now introduce the third aspect of my research, which was to evaluate prospective brain markers which predict later onset of mood disorders in high-risk individuals. So in one study, we found that a white matter tract of the brain, which is called the Usenet fasciculus, or short for UF, which prospectively predicted onset of bipolar disorder after an average of 6 years in high-risk offspring at an accuracy of 86%. So indeed, this white matter tract was the most significant predictive marker over and above the demographic and clinical questionnaire variables. In another study, we additionally found that the thickness of the cortex in the brain was prospectively associated with later onset of mood disorders among offspring of bipolar patients after an average of 6-year follow-up by directly comparing those who subsequently converted to full-blown mood disorder compared to those who remained clinically healthy. So these studies, they collectively provide new and supporting evidence for using brain features at early developmental phase of the high-risk individuals to achieve quantitative prediction of later disorder onset. So to summarize, we discovered a novel brain marker, which was the coupling between structural and functional connectivity of the brain, which contributed to the identification of bipolar disorder patients. We also found that the brain features related to the gray matter volume and its development during adolescence, which characterize high-risk individuals who are offspring of bipolar patients. And lastly, we demonstrated that the brain white matter tract and cortical thickness features prospectively predicted later onset of bipolar disorder among the high-risk individuals. So this is the end of the talk today. Thank you very much, and I'd like to hear your thoughts. Thank you.